Hey, so
Okay, well, Anna, let me ask you this. Hey, Anna. What I want you to do is I want you, when you're doing the reading and the on your own, kind of all along, I want you to be learning it all along so that you're not just learning it right before the test. That's why I do the participation grade to kind of help keep everybody accountable that they're working on learning it all along. So you don't have to know everything when you come in here, or understand everything, but you're at least showing me that you're learning it all along, because you're gonna retain the biology and it's gonna be easier to learn if you kind of learn it all along. But I think you're getting some along the way, more than you think. I think you are. Okay, girls. Okay, Essie and Ruby. Okay. All right. Okay, so Essie and Ruby, what did you think about the test?
It was better. I thought I did better than last week. Like, I feel more confident.
I didn't feel
as good, but I still felt pretty good.
it was the opposite for me.
I think Protista is one of the hardest. I told them that that's my least favorite unit that we study the whole year. So we're doing Kingdom Fungi right now. How are you liking that? I think you people that didn't do the mushroom walk would have liked fungi better if you had gone out and done the walk. I think that's like important. What did that kind of get you a little geared up about fungi?
I like mushrooms.
Okay. All right, so I'm going to, for the people who did bring in mushrooms, I'm not going to take points off for people who didn't, but for people who did bring them in, when I'm grading the lab notebook, I'm making a note of who brought them in, and there might be like an extra.
Well, I didn't bring it because I forgot to bring it, but I literally caught it and everything, and I was like grossed out because there were bugs in it. So I was like, I went through the torture of doing it, and then literally didn't bring it.
The torture, I actually thought, so I did it yesterday. I called one of my neighbors and went over to their house, and I didn't take my phone with me, and I was just reminded of how much it's just kind of nice to walk through the woods. It's just kind of peaceful and just looking at everything. I enjoyed it. Okay, everybody pull out your lab notebook. Did everybody get their table of contents caught up and get their first 3 labs caught up?
I got all of them.
Yeah, I got all of them.
Good.
We did it together on
Yes,
FaceTime.
we did. Okay. What I want to do is I want everybody, did you not bring your lab notebook today, Nancy?
No,
Did you?
I have it all caught up.
Okay. All right, I want to make sure I wrote down every lab we've done because Hannah was asking last time like what lab number we are on, and so I want to go through and make sure everybody's got their table of contents correct. Okay, so what is the first lab?
Using a biological key.
Yes, and that doesn't have to be the title. You could have named it anything, but it was the biological key lab, which is 1.1. What's the second lab?
Introduction to microscope.
Intro to microscope, something about a microscope, using a microscope, whatever your title was, 1.2. What's the third lab?
Pond hive.
Are you checking yours, Ruby, to make sure you got this listed? Not Ruby, Hannah. For some reason, in my head,
I
I
was like,
think,
is she talking to me?
sorry.
She's looking
Yeah.
at you.
Look at your contents and make sure you got this right. Okay, what's the third one?
Pond hive.
Pond life, 2.2. What's the fourth one?
Subkingdom protozoa.
3.2, yes. What's the fifth one?
Subkingdom algae.
3.3. Okay, and then we're gonna have 2 more today. So the 6th one is going to be the mushroom lab, which your book calls Phylum Basidiomycota 4.1, but you can call it something else. And then,
4.1? Okay. Yeah, mushroom. I'm gonna call it mushroom.
so that should be the 6th one listed. And then the 7th one listed is gonna be yeast and fermentation, which is 4.2. Okay, so that was just a guide. So everyone should have their table of contents completely updated, even you, Anna McBride. And your first 3 labs finished. If you have all of them finished or you've gone even further, that is wonderful, okay? So let's make sure you're getting that done. Okay, and everyone handed to me your test, your module study guide, and any corrections. If your corrections are separate, I think you gave it to me. Or
I
you
think
gave
I
it to me?
did, yeah, because it's not in there anymore.
Okay.
And it was before, so.
If you have something separate, yes, I have it. Go ahead and hand it to me. Okay. So we're gonna go ahead and turn in our books. We're gonna talk about kingdom fungi, and then we're gonna get into our 2 labs, okay? Unfortunately, we were supposed to have a magnifying glass for the mushroom lab. You do? Yes, thank you. I thought there was one in the box, and it wasn't in there, so that was a little disappointing, but that was wonderful, Essie, thank you. So, was that in your lab kit that you had?
Yes.
Okay,
It's like
awesome.
in
I was
like a little
looking in,
Ziploc bag.
I was looking in
Might
the,
be dirty though, so.
that's perfect. That's okay, we can clean it up. Okay, so we're gonna go ahead and talk for a little bit about fungi. And then we will get into the lab, and I'm actually gonna start the yeast lab while we're talking at some point. Maybe Eli, do you have a watch on today?
Yes.
Okay. Can you tell me, cause I won't look at mine all the time. Maybe like 12:35? Okay, all right, that'd be perfect. All right, so what are some characteristics about fungi? What do we know? What are some things that would put an organism in fungi?
Heterotroph.
Okay, let's raise our hands. Okay,
Heterotroph.
heterotroph. Okay, Essie?
Most are saprophytic,
Yes, Essie, what does that mean?
feeding on their waste of dead organisms.
Okay, who that did the mushroom walk found mushrooms that were not coming out of the ground, but actually on some kind of a dead tree or something like that? And maybe you didn't bring those, Anna, if you did, and Nancy. Were they on dead trees, or what, was it on something else?
We saw some, like, of the shelled ones on
Uh-huh.
a tree, but it was way too high, so you can't get it.
Couldn't get it.
Yeah, I've seen them before. I didn't see them because I just had some like literally right outside of my door. There's probably like 20 of like these
Oh, wow.
giant. Like I should have taken a picture because they were literally like giant.
Oh, cool, cool. Well, I don't know how you guys feel, but I think that the mushrooms are kind of pretty. Does anybody else agree with me? I
Some
figured
of them.
Anna Grace would give me a yes on
If
that.
it didn't have bugs in it, sure, but it was gross because the bugs.
So when I went on my little walk through the woods, there was actually a tree down. It wasn't a huge tree, kind of a medium-sized tree down in my neighbor's yard, and it had the shelf mushrooms growing all over it. And I didn't have my phone with me. I wished I had my phone so I could take a picture of it for you guys. But it was beautiful. Like, I mean, just these little ruffly brown things all over this. All over. I wish I could bring this tree into class, and I wish I had my phone. But I think they're really pretty. Okay, so we know that they're heterotrophs. They're saprophytes. They're going to feed on dead organisms. Now, Essie, are all of them saprophytes? What are the ones that aren't saprophytes? Do you remember that term?
Parasitic.
Yes, so they're going to be parasites. They're going to feed off of some kind of a living organism, okay? And so a lot of plants that get sick, like you might have some kind of a shrub or a tree in your yard that ends up getting sick and dies. A lot of times those diseases that cause plants to die, it's some kind of parasitic fungi. Okay, so they can be harmful. The ones that are harmful are going to be most likely to be parasites. Okay, so what else? Give me some other characteristics besides that. Nancy,
They have extracellular digestion.
what does that mean?
It's like they digest their food outside of their bodies.
And how do they do that? Do you
I'm kind of like still a little bit confused.
That's a good question.
know? Does anybody remember how they do that?
They secrete a chemical onto the food.
Yes, so they have some kind of chemical that's going to break down the food. They're gonna secrete that, and then that food is gonna be broken down into the nutrients, just the individual nutrient molecules. And then those molecules of the nutrient that fungi needs are going to be absorbed into the cells. So they don't have a digestive system inside their cells. Their digestive system digestion is happening outside. Some of those chemicals that they're secreting for that extracellular digestion are toxic. Not all of them are, not all of the chemicals are toxic, but some of them, that's one of the ways that they're toxic. Okay, what about where and how they grow? What do they typically grow in? Anna Grace, you remember what they grow in? And you can think about how they feed when you think about this.
Like fallen leaves and trees.
Uh-huh. So the key is they're growing in their food source. So when Anna Grace, when you found those shelf mushrooms on that tree, that dead tree or the dead limbs or whatever, the mushrooms are actually growing in their actual food source. The ones that are growing in the ground and the dead leaves, they're growing in their food source. Okay? And that's kind of how that extracellular digestion works, because they're growing in it, they release the chemicals, and then the nutrients come back in the cells. Okay, what type of cells do kingdom fungi have? What types of cells? Nancy?
Multicellular.
They're multicellular with what kind of cells? Remember, there are two kinds of cells.
Eukaryotic?
Eukaryotic. Multicellular eukaryotes, that's right. Is every organism in kingdom fungi multicellular?
No.
No, okay. Do you remember one that you studied, that you've read so far that's not multicellular? Anybody remember? It's in phylum, Ascomycota.
Which one?
The phylum Ascomycota? It's the yeast, okay? And I think, so some people might think, why, that yeast don't necessarily fit that well, because they're single celled. But yeast have something that all organisms in kingdom fungi have that no one's mentioned yet. And this is what I think why they're classified in kingdom fungi. Does anybody, the one characteristic, well, there's a couple characteristics that we haven't mentioned yet about kingdom fungi, but this is one that's true for everything in kingdom fungi.
Is that mycelium?
No, because they don't all have mycelium.
I underlined it in my book, but no, I don't. I like started it, but no, I don't.
Okay, it has something to do with their cell wall, and it's something that they have in common with some different insects and arthropods. Essie?
Is it spores?
No, but that's one, we will talk about that. No, it's a chemical that's in their cell walls that's different. Plants don't have this in their cell walls.
Probably wrote it down.
It is chitin, okay? Chitin, do you guys remember that? And the one time in this class where you'll hear about chitin again is that it's in some of the outer layers of the arthropods. It helps make up their shell because it makes their shell hard and protective but flexible. Okay, so chitin is going to be like in a lobster shell. It's going to be in some different insect protective coatings, but chitin is in, so fungi do have cell walls, okay, and they're going to have chitin in their cell walls to protect them. Okay, so let's get back to what Essie said. Another characteristic.
They don't form spores.
they can form spores, okay? And when they form spores, is that sexual reproduction? Does it involve mixing DNA from two different organisms, two different like organisms? Or is it asexual where it's the same DNA in the new organism and the parent?
It could be both.
Yeah, I think it.
Well, they can do both, but the spore is sexual. Okay, so the spore is gonna be a mix of DNA, and then they can do other kinds of asexual reproduction also. Okay? All right, so let's go ahead and look at page 98, the figure 4.1. Okay, and what is the, Eli, you had just mentioned this, but what is the largest part of the mushroom?
The mycelium.
The mycelium. So, did you, Anna Grace, did you know that the mycelium was the largest part? Since you like mushrooms, did you know that before this?
No.
Okay, so that part that grows underground, that's there all the time, where the part that comes up that we see that I think is pretty is the fruiting body. That's only there temporarily at different times, okay, and that's part of the sexual reproduction. But that part that's underground, that mycelium, it is 10 to 20 times the size of the fruiting body. So, you know, Bessie?
There's like one mushroom that, or not mushroom, but like a group of mushrooms that's like 2 to 3 miles long. My mom and I looked it up.
Oh,
That's
wow.
like the biggest one.
Was it just like a mushroom, or was it a fairy ring?
I think maybe a fairy ring. I
Uh-huh.
can't remember, but I just remember her looking that up
Oh,
because we were
wow.
trying to find different kinds of mushrooms we could find.
Uh-huh.
And
cool.
there was like one that was like 2 to 3 miles long. We're like,
That is, yeah, that's cool. Didn't you guys think the fairy ring thing from the video is really neat? I think that fairy ring's really cool. Okay, all right, so we know that the mushrooms, which are in phylum, what's it, how do you pronounce it?
Psittacicoda.
Thank you, phylum, Psittacicoda. So we're gonna have the mycelium is gonna be underground, okay, and then what we see above ground is the fruiting body, which we learn about a little bit later in the chapter. But it's going to be made up of the cap, the stalk, and then underneath the cap you have gills, okay? And then inside those gills, one of the things that we're gonna be looking for today, that's where the spores are gonna be kept. They're usually gonna be kept in some kind of a sac with the Basidium microbata. They're going to be in a club-shaped sac. And so we're going to be looking for those. Okay, so the mycelium, are those roots like a plant's roots? No. What are they, what do they do, Nancy? Remember? Sorry, sorry to put you on the spot, you were shaking.
It's like that, I guess they just draw the nutrients up from the
So, okay, so they provide support, so you think about that being 10 to 20 times the size of the part that's up underground. So it's providing support, you know, which roots, plant roots also provide support, but these are different. And then that is where that extracellular digestion is going to take place. So that's why the mushroom is growing in its food source. So that's those cells in that mycelium are going to release that chemical, and that's where that extracellular digestion is going to take place, and those nutrients are going to come into the cells. So the mycelium, what are all those little strands that make up the mycelium called?
hypha.
Hypha. And so you can see on page 99, you have two kinds of hypha. You have hypha that have clear distinctions between the cells, okay? And then you have hypha, so that's the septate, which is like a divided hypha. And then over to the right, you have a non-septate hypha, where there's not division between the cells, and you have all these nuclei, but they are separate cells, they're just not divided. The thing to know about the septate is, even though they're divided, the cells, those dividing lines, those cell walls have pores or openings in them, so stuff is still kind of going back and forth between the cells. All right. Okay, so we have different kinds of hyphae that do different jobs, okay? And so the type of hyphae that we were just talking about that's providing support and doing the digestion, does anybody know what kind of hyphae, what that is called? That's underground. So these are hyphae that are underground, and they're providing the support and the digestion. Eli?
Rhizoid hyphae.
Rhizoid hyphae, okay? So are all hyphae underground? And you can go ahead and turn to page 100. Are they all underground? No. What are the kinds of hyphae that are not underground called? Essie?
Aerial hyphae?
Aerial hyphae. So that is what, if you look at figure 4.3 on page 100, okay, you see the rhizoid hyphae are down in the ground. And right now, this one does not have any kind of fruiting body. Those things that are coming up in the air with the little puffy balls things on the end, those are aerial hyphae, okay? And they're going to absorb oxygen, produce spores, and they will form new filaments with a form of asexual reproduction. Okay. All right, so also on this diagram, you're going to see the stolon, okay? And the stolon is a type of aerial hyphae that does the asexual reproduction. So that stolon grows off of the one fungi, and then it grows, it's above the ground or above the food source, and then it's going to form another one that's going to be identical. So those two next to each other are going to have the same DNA. That stolon asexually formed another hyphae, another organism with the exact same DNA, okay? And then in the tops of the aerial hyphae, where they produce these spores, you're gonna have, those are called sporophores. And the sporophores are spores that are covered, and when they're not covered, they're called kinetophores. All right. And you can see right on page 101, chitin, the chemical, that's what's in the cell wall. It provides toughness, but flexibility, okay? So there's cells, so you know a mushroom is not, it's not gonna be super hard. You can cut through it, okay? So that chitin is gonna make it tough and give it protection, but it's still gonna have flexibility. All right, let's see here. A fungus produces Haustoria, which is, what was the Haustorium? Did I skip that? Does anybody remember what that was?
It's on page 100.
I see it now. Okay, the kind of hyphae is an extension of the mycelium is called a Haustorium. A Haustorium is the hypha of a parasitic fungus that enters the host cells, absorbing nutrients directly from the cytoplasm. So, a fungus that produces Haustoria, is that saprophytic or parasitic?
It's Paris.
Parasitic. Okay, I'm looking right on your own. Okay. A farmer tries to remove a patch of mushrooms from his field by pulling up all the stalks and caps in the patch out of the ground. Why has the farmer really not gotten rid of the fungus? Nancy?
Because the mycelium is still there.
The mycelium is still there. That was, he only removed the fruiting body. Okay. All right, with the spores, how are these spores spreading out to produce new fungi? Anybody remember how the spores are getting out? I guess the one video that you guys watched with the puffball, did you like that when they went and kicked the puffball? Did anybody see any puffballs when they were out? Last year, my class said that they had puffballs. Different people had seen puffballs. I don't know that have you seen a puffball before, Anna?
I think I have, and it was like the size of that, and
Oh,
I kicked it.
Oh, did it release the spores? Did it release?
I'm not sure. I can't remember. It was like a couple weeks ago, but it was just growing in my yard.
Okay, well, everybody also, if you, this school year, even when we're done with fungi, if you see some really cool thing like a puffball or a slime mold or something like that, please bring it in, even if we're past this unit. I think it'd be cool to see. But Anna Grace, have you seen one before?
I've seen puffballs before, but it's been a long time.
It's been a long time.
But they are edible.
Oh, they are edible? Oh, really?
Yeah, they're not, if you eat them, you can cook them.
Are they good?
Just gonna eat it.
Oh, well, that is very
It's
interesting.
a giant puffball.
You should bring us in. You should consult.
Okay, so we're gonna look on page 100 now. So the phylum that our traditional mushrooms are gonna be in are the Basidia micata, okay? And those have spores in their gills, and they're encased in a club-like little bag called the Basidia, okay? And then we also are studying Ascomycota. Those spores are going to be in a round sac, but the yeast are also in the Ascomycota, okay? And then these are the 6 phylum that are listed here in Kingdom Fungi, okay? The last one I wanted to note to you, the Myxomycota, it says fungi that look like protozoa for much of their lives. If any of you listened to the little Taylor Swift 22 song that was on my the review song, did y'all listen to that? You didn't listen to it? Okay, well, it said something about fungi like protist, okay? And one thing I want to tell you is that we're classifying the fungi like protist in Kingdom Fungi, but a lot of scientists do not do that. They actually classify them in Protista. And so some people, depending on your science textbook, or the scientist who's ever written your textbook, you're going to cover that phylum in Protista. But for our textbook, it's considered part of Kingdom Fungi. Okay, so that figure down there on the bottom of page 103, that's a picture of the puffball. Also the shelf fungi, which, Anna, Anna Grace, is that what your shelf fungi look like?
Shelf fungi.
Did they look like that type or not?
They're smaller and they're different. I think I'm not sure which ones I have. I think I have like a turkey one, but I'm not sure what.
Okay. So you did you go ahead and get your book out and try to identify some of
I
them?
haven't had time to
Okay.
do it. I was going to.
Okay, that's okay. You didn't have to do that. Okay. All right, so let's go ahead and look on page 104. And this is going to be the life cycle of a mushroom, okay? And so I want you to start, and at the, kind of that left upper corner where it says 2 mycelia growing from spores. Okay, so you have 2 spores from 2 different original parents, okay, that have good conditions, and so they have started to grow into mycelium, okay, which there it looks like those mycelium are made up of what type of hyphae in that diagram on page 104? What type of hyphae is that? Septate or non-septate? Nancy? Septate. So you got 2, because they've got the distinct cell wall, so it's going to be septate hyphae, okay? And you've got 2 different ones that have grown from 2 different spores. So this is 2 different organisms, okay? And you can see that they're kind of crossing each other, okay? And they're going to fuse and exchange DNA, okay? So this is why it's sexual reproduction, because you've got 2 different distinct organisms, okay? And so then, when you go to that kind of top photo, the mycelium formed as a result of the reproduction is going to grow quickly, and it's going to crowd out the 2 original parents. So the new offspring is going to grow quickly and crowd out the original parents. All right. So then you go to that mycelium is going to form kind of a membrane. Then it's going to be in the button stage, okay, where it's going to start, the stipe and the cap are emerging from that button. And then you're going to have what we got to pick out of the ground, okay? So you're going to see the full stipe, the cap, you're going to have the gills. And then when you get closer up to the gills, what do you see? What is that? When you, in that picture where it's close up of the gills, what are we going to look for in the gills today? Nancy? And what are they in?
Basidium?
Basidium. So those are going to be the little club-shaped sacs that the spores are in. And then those basidia are going to release the spores. And then look, we get right back to the spores are then going to form hyphae. And then those hyphae are going to fuse. And that's kind of how that life cycle is going to go around. Okay. The toxicity. So we know that mushrooms can be toxic from the chemical that they release for extracellular digestion. Is there a way for a normal person hasn't studied mushroom morphology and everything about the mushrooms to be sure that they're not eating a poisonous mushroom? Is it a good idea to eat mushrooms out in the wild? No. Okay, so it's always safe to get mushrooms from the store. And there are people who are experts in that, but that's never, ever a good idea. Okay. All right. So besides the puffballs, fairy rings, shelf fungi, those are all in Basidia microta. There were 2 that were in Basidia microta that the book talks about that can cause problems or damage to crops. Does anybody remember those? Those are mentioned on page 107. Essie?
Is it wheat fungi?
So it does hurt wheat. So one is called a rust. Okay, and this is going to look like a rusty colored something on the leaf of something. So it attacks wheat. And the other one is a smut. Sorry, Ruby. Okay, so both of those are going to be parasitic fungi, and they are going to be bad for the plants that they're growing on. One of the things I wanted to mention with these parasitic fungi, a lot of times, and the rust is like this, it has to have what's called an alternate host, okay? So the wheat is a host, the fungi, the rust is a parasite, okay? So the parasite feeds off of the host and damages the host. But that rust has to have, it can't be on the wheat all the time. It alternates between the barberry bush, which is their alternate host, and the wheat. So it survives on the barberry bush and produces a spore that then goes to the wheat and produces a different kind of spore. And so they have to go back and forth. So you definitely want to make sure that you don't have any barberry bushes growing anywhere near your wheat so that you're not providing that alternate host. Okay. All right. So with the lab that is 4.1, the Basidia micata, we've got a lot of mushrooms out there, different kinds. And there's shelf fungi, so that would be in Basidia micata, and multiple different kinds of mushrooms. There's a little piece of what I think might be a slime mold, which we didn't talk about, that I stuck out there. But I think everything else is probably in Basidia micata. And so you're going to pick a mushroom, you're going to get a plate. I've got a bunch of dissection tools out there. Essie brought hers. Is that what that is? No.
No, I forgot.
Oh, it's fine. It's fine. We got enough stuff. And so you can, I want you to examine it. I want you to use Essie's magnifying glass and try to see if you can see the little basidia up in the gills. Okay, you're probably gonna like cut the mushroom apart. And then you're going to attempt to make a slide with one of those basidia with the spores in it. And we're going to do, what we'll probably do is everybody can practice making a slide, but we'll probably just pick two that we'll put on 2 microscopes. And so you will draw and describe your mushroom, okay, from looking at it on the plate, there are plates out there, and from cutting it apart, kind of what you see. So it's gonna be maybe a drawing and some descriptive words. And then, so this is all gonna be in your data section. And then after that, you will draw and describe what you saw on the slide, okay, and what magnification you have put it on the slide. Does that make sense? So that's what we're doing with the mushroom lab. All right, Eli, you forgot.
Oh.
Okay.
It's still 36, it's still.
Okay,
It was a
so
minute.
then I'm gonna talk for just a few more minutes about the yeast, but I'm gonna get this started. And so we need a tablespoon of sugar and 2 cups of warm water. All right, so I'm going to put my sugar in there, put my water in here. Stir that around a little bit. Okay, so these are, yeast are gonna be single cell. They're in Phylum Ascomycota. And the reason that they're in kingdom fungi is because they have chitin in their cell walls. And those are yeast spores, okay? So the yeast, they get the yeast spores. And now because we're putting it in warm water with a food source, the spores are gonna become yeast cells. And they're going to feed off of the sugar, okay, that's in there. And does anybody know what that yeast is going to release? You would have seen it on your video. You guys have already watched this video, haven't you?
No, I forgot to watch it. I was gonna watch it, but then I forgot.
Okay, hey guys, we gotta get on it. Y'all definitely, I mean, the one thing I expect when you come in here is that you've done the reading, the questions, and watched all the videos, okay? All right, that's okay, but thank you for admitting it, because you're probably not the only one, okay? But let's make sure Eli, I think he's given himself up to, okay? So I spend a lot of time looking for those videos, okay? because I can't teach you everything and I want to show you things. And so to me, the video should actually be kind of the fun part. Did you guys watch the video about how to tell the mushrooms are poisonous? Okay, that was a really good video, Ruby. It shows you the fairy rings. It was a really, really good video and it was fun to watch. So I want you to promise me, and anyone else who didn't watch the videos, that you go back and watch the videos. Sometimes I put questions specifically from the videos on the test, and if I put a video there, I mean, there's a reason. I'm not just gonna give you a video to watch. So please, please, please make sure that you're watching the videos. It's important. Okay, so the video will show you that this yeast, what is it releasing? Who did watch the video that remembers or got it from the reading? What is the yeast? What is gonna happen? What are we gonna start seeing from this? Anybody remember?
I don't remember. I watched it, I think I just forgot.
Okay, it's okay. It's okay. So it is going to release carbon dioxide. Okay, that's what the air bubbles are going to be. So we're going to start to see this bubbling up. Okay. It's going to release carbon dioxide and it's going to release alcohol. Okay, so you might be able to smell some alcohol smell. So this is called, does anybody remember what this process is called?
Fermentation.
Fermentation. Okay. All right. And so on this lab, which is in your book on 110 and 111, okay, you're going to describe what we see going on in the bowl. In a few minutes, when it kind of gets going, I'm going to have everybody come up here and look at it. And then we're going to make a slide, okay? And we're going to look at the yeast under the slide. We're going to do one slide without the methylene blue stain and one slide with the methylene blue stain, okay? And what we're hopefully going to see is a yeast cell budding, okay? And I actually included in tomorrow's work, there is a video of a yeast cell actually budding, because I wanted you to be able to see that. And so what's going to happen is that it's going to be very similar to kind of like what we studied with the amoeba, and that the cell is going to replicate the DNA, and then a pouch is going to kind of form a pouch out to the side. That extra DNA will go into that pouch. It's going to draw some cytoplasm into the pouch, and then it's going to form its own cell. So what you'll be looking for on the slide is kind of like a cell with a pouch coming out to the side. And then eventually they're going to form colonies, so you're going to see long strands. So you might even have long strands kind of budding together. So what I'm hoping is that you're going to see that on the microscope today. If we don't get to, I think we did last year. I think it was successful last year when I taught it. But if we don't, you'll get to see a very good image of it on the videos. And I want everybody caught up on videos this week, okay? The other video that I added for tomorrow is a video from the microscope, and it's of spores. And the reason I did that was because I don't know how the lab will go today. I don't know how our microscopes will do and how well we'll see the spores. And so if yours was not a success, you will have that also to look at, so you can kind of see what it should have looked like. Okay, so a little bit of bowling going on, not completely. So I'm going to let everybody go ahead and go out to the table and pick out the mushroom that you want in a plate. Okay, Anna, I'm going to turn it off for the lab.